Cool. Thank you so much for inviting me, and thank you for the lovely introduction. Very excited to talk to you today about my work, which is joint with Fabian Spool and Hong Duong. Oh, okay. So today I'm just going to start with a biological motivation for all of this, which will ground the models I talk about next in the background, which is really what we directly built off in our own work. And then with that context, I'll finally talk about our results and then give a conclusion. So biological motivation. Okay, so sort of the whole theme of this research can be summarized by this quote here. It's not the strongest of the species that survives, nor the most intelligent, but rather the most responsive to change. So what this is really saying is it doesn't really matter how impressive or well adapted the species is to start with. If it can't evolve by natural selection, it dies out. And we're going to flip this quote a bit on its head by the end of the talk. But let's talk about like an example, so a biological example. So this is really what the picture that sort of inspired most of this work. We have, this is taken from a biological paper where they give a hypothetical phenomenon in cancer biology called decoy fitness peaks. Now, to explain what that is, I need to talk a bit about, I need to explain this image. So sort of on this X here, the genotype of the cell, which is just what genes it has and whether or not they're mutated. So beneath this sort of main peak is WT or wild type cells, which is just normal cells, unmutated. They're doing what they're supposed to do. And on the Z-axis, you have fitness, which is just a measure of how well they reproduce and survive and don't die in that environment. So in the young organism's environment, the wild or normal cells, or they're the fittest, which is kind of as you expect. That's good. But then you have these two other peaks where these cells that have like some certain mutation in a gene called Notch, and you have other cells that have a mutation in a gene called TP53. And so what you should know is that TP53 is sort of the most well-known pro-cancer gene. So it's bad, right? So in the young organism, though, it's fine because you have all these normal cells and they're going to outcompete those because they're the ones that are best surviving. But then in the middle-aged organism, something interesting happens. So this normal cell declines in fitness, but both the cancerous and non-cancerous mutants increase in fitness. And so the whole point of this paper is that they're proposing the existence of this secondary peak here. And because this exists, the cancerous cells will have mutation and therefore be suppressed. So it's kind of like an unconventional idea is that actually having some mutations is cancer suppressor. And sort of or fitness landscape is changed way through time. And this slide is just to say it's not completely theoretical. People have shown this more or less happens in mouse models. So this is from another paper, again, not by us, this is by experimentalists, where they sort of seed a mouse with various microtumors. And then they also cover it with a certain proportion of it with competitive non-cancerous mutants. And the more non-cancerous mutants they have, the more microtumors are eliminated. So like this actually does happen in mouse models, whether it happens to people or not. But I think this is enough to say, okay, let's start to think about how we can even start modeling something like this. And so that brings us to the model. So you can do this in lots of ways. You could do like a stochastic model. I'd like PDs, so I'm doing a PD model. So let me explain a bit what's going on here. So n of xt is a number of individuals with trait x at time t. So the trait x just tells you where you are on that fitness landscape that I showed you just now. And there's individual things, right? Which is represented by this epsilon squared Laplacian term. And this is just to say mutations are like very local. You can do other stuff, but this is a very common one. And they can also reproduce an exact copy of themselves, and they'll do this at rate a epsilon of xt. And so a of xt is going to give this exactly the fitness landscape I showed you. So it evolves through time. Competition of every other phenotype. So it's just logistic growth, but the competition is between everything. Okay, and then you have a boundary condition and initial condition. So I'm going to now go through some work from other people that we built off directly. So you could start with the case that this is actually time independent, so you're not considering a changing environment. But what these people did is they considered having multiple peaks of this function. So maybe I could even, does the recording capture the whiteboards?
But if it's small, just go for it.
Yeah, I mean, it's just so you have a picture. You'll have something anyway, so it's just like that, right? Something like that. You have multiple peaks, or I'm really saying. Okay, and how do you go about analyzing something like this? So this is going to be kind of the common way to analyze every single model I talk about. The details will differ, but this is the general, the eigenvalue problem associated to it. And say a couple of things. One, you can say that a positive exists, and this is important because it's a biological application. You don't want a negative number of cells. Going to converge to a multiple of that eigenvector in the long time. So we understand the eigenvalue problem, we understand the long-term behavior of the solution, and then you can get exactly these limits for these two, the eigenvector and the eigenvalue. So as this epsilon, which is the mutation rate, goes to 0. And so as the epsilon goes to 0, our eigenvector is going to converge to a sum of Dirac deltas on some set S. Now you might guess that the set is exactly those peaks of the fitness function, right? It's survival of the fittest. And indeed, that's what you get, but the people in this work went a bit further, and they said you actually converge to the flattest peak. You will have a Dirac delta on the flattest peak. So this is for like a finite but small epsilon, you have something that looks like this. And that's just, this is the L1 norm of the eigenvalues of the Hessian at the peak, which is just a measure of flatness. And that's you concentrate on something that minimizes this, but maximizes the function. Okay, but we really want to start thinking about time-dependent ones. Just about one more previous model that will give context to ours. So these people went for a shifting and a fluctuating environment. So E of t is a periodic function, and in the x variable we now have like x minus the epsilon of t. So the whole thing is shifting periodically, is moving in one direction, but also fluctuating periodically. In the end, we don't really care much about the periodicity because in our model we don't use that. The important thing is these assumptions here. So they assume that the average has only one maximum, and they assume that that if you go far away enough from this maximum, that it's going to be negative. So right, like there's some optimal phenotype, and if you're not near that phenotype, you're going to die out. And lastly, that there's some x bar that's this equation here, and the limit as epsilon goes to 0. So the reason it's called the lagged optima is because the optima is here, but you're just kind of lagging behind, and all this is kind of shifting on in the direction, CT. Okay, what our work now. So we take this form of, so it's a bit different in this way. So we assume we can decompose A into two functions that are going to be shifting at possibly different speeds. Without loss of generality, I can say C1 is less than or equal to C2. And we assume each of these is compactly supported. And remember, there was this assumption that if you're far away enough from an optimum, you're negative. Compact support and the addition of this minus delta term here will make sure that you have essentially the same requirement there. And then for each AI, we put essentially the same assumptions that had we put on the average. That is, we assume for each AI, there's a unique lagged optima, which are just numbers that satisfy these equations here, right? That again each one depends on the maximum of its respective function, A1 or A2, and on the shifting speed of that shifting mass. Here's a numerical illustration of this. Will I be able to get this on YouTube? There's a YouTube video linked here. Let's hope it works. Yeah, thank you. Yeah, so you can see over time it started off kind of symmetric. It's concentrated on both, and then eventually only concentrated on one of them. So you can't really see it here, but the peak where it concentrates on is in fact the flattest of those two peaks, as you might expect from the results of the other paper. Could you close the YouTube now? Thank you. Right. And so what were we able to prove in this regards? Well, we could go as far as saying, okay, there is some time-independent positive eigenvector solution to the associated eigenvalue problem, right? Because that's kind of how we're always going to analyze this. And that this eigenvalue vector is going to concentrate on the set of lagged optima. That is, it's going to concentrate on both of them. But from what we just saw, one or more of these AIs might be zero, right? Because it was in the end. Oh yeah, and that's just saying, okay, the long-term behavior, it will concentrate to multiple of this. We couldn't get quite so far as showing that it would concentrate on the flattest one, but what we did do is you can consider this other eigenvector problem, and you get that the solution to this will just concentrate on the lagged, on the flattest optimum. And this turns out just to be like a weighted rescaling of the eigenvector that the solution actually will converge to, a multiple of. And so from this, you sort of expect that result. You can only really get that in particular cases. For instance, if p epsilon were a Gaussian, you just kind of do, completing the square and the exponent of the Gaussian, and you kind of see only the one that is following the flattest optima is the one. So there's partial results there. But let's talk about the second case. In the second case, does not equal C2. We have to make, some standard assumptions, which I'm not going to bore you with. They're just, you assume, both A1 and A2 are sort of smooth enough, and you have a nice initial condition. But the important assumption is that you assume this lagged, one of the lagged optima is greater than the other, and it's positive. So this is just a short way of saying, okay, there is a maximum positive lagged optima. And that's exactly this value here. In that case, here. So there's some function that's going to tend to, like, if you shift time far enough in the future, you get convergence to just the maximum of the positive lagged optima. So we know exactly where this is going to concentrate over time, depending, again, both on the maximum and on the shifting speeds. So illustration of this. Symmetric initial condition. I fix C1 as -1 and C2 as 1. And so this one, so the sort of, the A2 function is shifting to the right, A1 function shifting to the left. And it concentrates on the A2 function, because I happen to have chosen this in such a way that it has a higher maximum. And then as I shift C2, this lagged optima is going to decrease, because I'm taking away C2 squared. And eventually, it's small enough that we start concentrating on just the lagged optima following A1, right? Because now this has the maximum of the positive lagged optima. I also have a video of that, if you could click on. Thank you. Okay. You can see it sort of initially concentrates following that one and lagging behind, but then it sort of can't keep up with it, or it can to an extent, but now this one is going to take over. So we now know, yeah. Okay, thank you. Right. Okay, so that means we actually know in that case we know a bit better than the previous case. Just to conclude, I think this actually went a lot faster than I thought, but we got a solution to what's sometimes called a selection mutation model with multiple shifting optima, and what I've just showed you is that the lagged optimum, the subpopulation which has the highest positive lagged optima always wins. I've shown that if they're the same velocity, you set of lagged optima, which is new compared to the case where you don't have a shifting environment. And we can almost show that it converges on just the lagged optima following the flattest of those peaks. And this is applications to competitive interactions between cells in aging organisms, or to animals surviving climate change, where the environment is shifting, and yeah, I guess, as I said, we're going to flip that quote on its head. Here is perhaps the new quote: that it's not the strongest of the species that survives or the most intelligent, it's that which requires the smallest response to change. So in other words, if you change some variable in the environment, not necessarily everything's going to be affected in the same way. And that's kind of as important to figure out which one's going to survive. Questions, comments, collaborations, feel free to reach out to me at the email address here. Thank you.